Good evening, ma'am.
Good evening, sir. Sir, you can start now.
Yes, ma'am. Yes. Good evening, my dear students. So you are all good evening, Ajay. So you are all are warm welcome to the once again the biology class. Last class, what we discussed already, you know, about transcription. Okay. And the transcription process, what are the main three event is the that is called as initiation and elongation, and third one is called as what is the terminations. Okay. So each and every process, what are the requirement is the how it will be synthesized DNA from DNA to RNA. Okay. So RNA synthesized from the DNA molecule already in our laboratory with the help of the RNA polymerase enzymes 1 and 3 with rho factors. All those things already we discussed. Okay. So in this class, what we are going to discuss about, you know, what are the main important point of the transcriptions? What is the main important point of the transcription process? The first one is the promoter is present at the 5' end of coding stand. This is first point. Promoter always present in the 5' end. Okay. So the process of the transcriptions takes place from 5' end to 3' end. So this is the first point. This is the first important point. Second one is the movement of the RNA polymerase requires DNA strand rotation and topoisomerase and helicase activity. What is the meaning of that one? You know, when the RNA polymerase move on to the, see that, when the RNA polymerase move on to the stand 5' to 3', see that the promoter region present the 5' region, isn't it? First one. So during this process, that polymerase move on to the DNA stand. What are the three enzymes is very, very essential, you know. Two enzymes have been reported. What are they? Topoisomerase and helicase. So these are the two is very, very important for the DNA rotation. So then only the RNA molecules also, how it will be helical structure into straight in manner. Then only the, what is that? The movement will be very fast. That is a process. Okay. And then third one is the sigma factor recognize the promoter and a direct core enzyme to promoter. Beta and beta dash subunit form a crab claw like a structure with grips DNA. This is a crab claw. It can move slowly, just like a crab in the DNA stand. Okay. So then only the RNA molecules production takes place. So these are the 4 points is very, very important for the transcription process. What are the process? Promoter in the 5' end, RNA polymerase require DNA stand rotation. With this, topoisomerase and helicase activities. And third one is sigma factor recognize the promoter. Already know very well about it. It is the initiation point along with the core enzyme. Okay. And beta dash subunit form a crab claw like a structure which grips DNA. It can be easily bind the promoter region. That is very, very important on the DNA strand. That is very, very important. So these are the 4 points have been reported. See, the next one is reverse transcription and termination. So already you studied. What is it by reverse transcription, Ajay? Reverse transcription. Transcription means?
So transcription is when it's from DNA, when it's when RNA is. So reverse transcriptase is from DNA RNA, sir.
No, it's not correct.
So like the other way, sir. Sir, in retrovirus and all, in AIDS and all, it happens, sir. Reverse transcription.
Okay, good. What
sir,
I'm asking, yeah.
Sir,
Right,
it's
yeah.
basically when from DNA, sir.
Okay.
Usually RNA is converted into DNA, right, sir, in transcription.
No, that is reverse transcription, isn't it?
Yes, sir.
Transcription, now only we are studying DNA to RNA. So transcription. RNA to DNA, and then yeah, RNA is called as what is that?
Yes, sir.
Reverse.
Sir, reverse transcription, sir. That's what I
Okay.
said.
Okay,
Yes, sir.
okay. Good, good. Clear. Are you clear now? Yeah. Okay. See that a reverse transcription is otherwise called as terminism. Okay. Some viruses such as HIV and COVID have the ability to transcribe RNA into DNA. See that they are having the nucleic acid genetic material as an RNA molecule. When the RNA molecule produces the DNA molecule, that process is called as what is it? Reverse transcription. When the DNA molecule produces the RNA, that is called as transcription. Okay. When the reverse transcription takes place, what happened? One of the most important enzyme is also very, very essential. That enzyme is called as what is that? Reverse transcriptase. Without reverse transcriptase, what happened? The RNA molecule will not produce the DNA. Okay, see that the main enzyme responsible for this type of transcription is called as reverse transcriptase. Enzyme is reverse transcriptase. Process RNA to DNA. Okay. In the case of HIV, the reverse transcriptase is responsible for the synthesizing a complementary DNA strand that is simply called as cDNA to the viral RNA genome. Okay. This was discovered by Temin and Baltimore. So the name of the scientist, the reverse transcription was first discovered by, what is that? Who is that name of the scientist?
Sir,
Temin.
Temin and Baltimore.
Yes, very good. Exactly correct. Note it down, my dear students. This is very, very important. Okay. Sometimes they may ask the question, reverse transcription process or reverse transcriptase enzymes also, who discovered? Like that, they may ask the question. So please see the entire structure. Simply, we may call it, this is called as, this is the double stranded DNA molecule. Okay. When the double stranded DNA molecules itself produce the exact copy of the DNA molecule, that process is called as replication. That is, DNA to DNA is called as replication. When the DNA molecule produce RNA, that is called as transcription, first reaction. Okay, DNA to RNA is called as a transcription. Then RNA to DNA. See that lower, there is a red color arrow user and also small star-like structures, also indicating. Here in the last corner, slide last corner, reverse transcription. That is denoted as RNA to DNA. That is called as reverse transcription. So RNA to DNA is reverse transcription. Okay. Fourth part is DNA, RNA, and protein biosynthesis. This is called as translation. With the help of the DNA molecules and also the RNA molecules. What are the RNA molecules? There are 3 RNA molecules essential for the translation process, otherwise called as protein biosynthesis. We will see the next part. So RNA, so you will get protein molecule. This is called as translation. This is very, very important, my dear students. Okay. So this is collectively called as central dogma. Central dogma theory. Okay. So don't forget. Already we studied it. Once again repeated here also. Okay. See that. DNA to DNA. So that is called as replication. DNA to DNA. DNA to RNA. Transcription. DNA to RNA. Transcription. RNA to DNA. Reverse transcription. Because it's a transcription, it is reverse transcription. DNA, RNA, and protein, that is called as translations. Okay. This is also a small explanation. Otherwise called as what is that? Just like equation aspects. That is depends upon the structural aspect has given here in this part. What happened, they have given in the equation aspects. I think this part is completely finished. So next one is, we will pass on to the next one is question part. Okay. Just see the question, you answer it, my dear students. So the first one, the process 1 and 2 and 3 in the image respectively are. Now only we studied, isn't it? Question 1. See that number has given here 1. DNA, and it produces the DNA molecule. That process is called as, what is that, Ajay? DNA produce DNA. That is
Sir, replication.
very good. Okay, DNA to RNA. DNA to RNA. DNA to RNA.
Sir, transcription.
Okay, so RNA to,
Sir, RNA to DNA reverse transcription.
okay, here what they have given is, just see the slide. Here they have given us a 2, isn't it? So DNA to RNA comes. And then RNA to protein. RNA to
Sir,
protein.
translation.
So which one is correct in this part? A, B, C, and D. Which one is the correct one? Which one is correct, my dear?
First is. Sir, is it? Sir, is it? Sir, is it C, sir? Replication, transcription, then translation.
Here
Option
you
C.
take. Okay, very good. Exactly correct. You are taking too much of time in this part.
Sir,
Okay.
no, sir, like the diagrams are just checking,
Okay,
sir,
okay,
cross-checking.
okay. No need to worry. Simply you have to identify it within few seconds. You have to answer it. Okay. Okay. Anyhow, it will take some more times. Better understanding. So this is a replication. See the next one. You can have the solution also. So not it. Yeah. Okay. It's easy one. That's why. So here you already know about this part. See that one. DNA to DNA, isn't it? So this one is called as a replication. DNA to RNA transcription. RNA to protein bias translation. That is the thing. This is also already asked the question in NEET part. See the next one. Which of the following RNAs act as a structural RNA as well as ribosome in bacteria? Okay, already we studied this part fully. In bacterial DNA, okay, only 3 parts only present except one. Which one? Will you please? Excludely, like that they asked, isn't it? The question. Do you know that answer this, my dear? 5s, 18s, 23s, 5.8s. See the answer?
So 23 years.
So yes, the solution is C, 23s. Because 23s RNA, the Swedberg RNA, this is S denoted as Swedberg unit. And also another one is called a sedimentation unit sometimes, or RNA is component of larger subunit of ribosome. And it acts as a peptidyl transferase, that is called as ribosome. Okay. So answer is, what is that? 23, yes. So this one is correct. These are all or not comes. Suppose we are adding these two, you will get the third one. So 18 plus 5, you will get 23. So you must note it down. 23s is very, very important. We will see how many molecules present in the rRNA. We will see the next topic. And what about the third question? See that one. The cutting of DNA at specific location become possible with the discovery of cutting. So what is by ligase? Ligases, you know about the fragments of the DNA molecules. That is called as Okazaki fragment, isn't it? They link together with the help of the enzyme DNA ligases. Restriction enzymes, already you know about it, is very, very important ones. The restriction enzyme is act as a molecular scissor, props and selectable marker. Okay, these are all not comes. These two, it will not comes. Already you studied that. You didn't study these things. These two only already you studied, isn't it? So these two only the answer. Okay, the ligase it will not come. Already I told you. The fragment only attached with each other with the help of the enzyme ligase, linking of the DNA strand. That is Okazaki fragment. So RNA enzymes, that is called as a restriction enzyme, is very, very important. See the solution. The answer is the restriction enzymes, because the restriction enzyme is essential for the A cutting of DNA at a specific location. The specific location is called as what is that? Polyandrome. Okay, become possible with the discovery of restriction enzymes called as molecular scissors or molecular knife. If you want to cut any place of the DNA molecule, you need an instrument or knife or scissor. It is the molecule cut with the help of another molecule. Okay, that molecule is called as what is it? RNA polymerase. Otherwise, sorry, DNA, a restriction enzyme. Okay, restriction endonucleases. That restriction enzyme is called as what is that? Molecular scissor. The molecule cut with the another molecules. One molecule cut with by the another molecule. That enzyme is called as what is that? Molecular scissors, otherwise called as knife. It is very, very important, my dear. Molecular scissors also called which one of the following enzyme? Restriction enzymes. Okay, don't forget DNA restriction enzymes. Which of the following is not applicable to RNA? Which of the following is not applicable to RNA? Chargaff rules. What is meant by Chargaff rules? My dear students, one purine molecules combined with the molecules of another pyrimidine.
Pyrimidine, sir.
Yeah, good. Number of purine molecules equal to number of pyrimidine
molecules.
Yes, both are equal. Okay, double strand and a double hydrogen bond, triple hydrogen bond. Adenine combined with the molecules of double hydrogen bond and guanine combined with the molecules of triple hydrogen bond. So these are all are the Chargaff rules. It is present in the DNA or RNA. Suppose, take an example, when the two strand is present, where it present? Two strand is present. Where the two strand is present? Only the DNA molecule. So the DNA molecule only applicable Chargaff rules. Okay. Here what they ask the question? Not applicable. Okay. Chargaff rule is the first one. Okay. It is a correct answer maybe. Complementary base pairing. Complementary base pairing already in our board. It is also present both 5-phosphoryl and 3-hydroxyl. It is also present in both. Heterocyclic nitrogenous base. It is also present in both. So these three it will come to the RNA. The Chargaff rules only applicable for DNA only. So the Chargaff rules it's not come for or applicable for to the RNA molecule. See that result? The solution, Chargaff rules is applicable only for DNA, not for RNA. So this is the answer. Okay, don't forget, my dears. This is also important. And the last question, I think 2 questions more. I think transposons can be used during which one of the following? Gene sequencing, polymerase chain reaction, otherwise called as PCR, gene silencing, and autoradiography. Okay, autoradiography, it will not come. Polymerase chain, it will not come. Okay, so these two only, particularly, but what is by gene silencing? What is by gene sequencing? Gene sequencing is nothing but the arrangement of nucleotide, isn't it? Which one it comes? Maybe, see that one, gene silencing. What is by gene silencing? See that the gene silencing is the regulation of the gene expression in a cell to prevent the expression of a certain gene. Such type of gene is called as gene silencing. One gene suppresses the another gene. Already we studied. Okay. So here is also very, very important. So gene silencing can occur during the either transcription or translation. See that one? What the question? Transposons can be used during which one of the followings? Gene silencing also called as what is a transposons. Already we discussed. So other threes it will not come. Gene silencing is the correct one. What is the last one? The silencing of mRNA. Okay, this is it's out of syllabus only. out of syllabus, not only out of syllabus, it is also an examples has been used in producing transgenic plants. Here they asked the transgenic plant. Which RNA? mRNA has been used. That means isolated from the any one of the organ. Which one of the organ they told? See that white rust. White rust means it is also one of the microorganisms. Bacterial, it is also microorganism. Ball worms, it is also one of the microorganisms. Worms, so it's also organism, animal sources. Nematode, it is also a animal source. Particularly, the mRNA is isolated for the production of the transgenic plant resistant to nematode. Okay, so we will see this one, transgenic plant. Anyhow, just one questions extra from this part. So up to this, we will finish it. The answer is nematode. So the molecular base of inheritance part 5 is finished. We will pass on to the next one is part 6. Okay, my dear. See that one?
Yes, sir.
Okay, good. See, the part 6, what we are going to discuss? Any guess, my dear? Any guess? See the part 6. So already we studied DNA structure. Okay. And the part 6, we are going to discuss suppose that RNA and types of RNA. Okay. Already we know our body, Ajay, isn't it? How many types of RNA already you studied, my dear? How many types of RNA?
So mRNA, tRNA, rRNA, then there is hnRNA, then snRNA.
Very good. Exactly correct, my dear. Super. Nice. Okay. The RNA is divided into 3, already you know about, but here, so the mRNA is the messenger RNA, already you told. ncRNA is also there. Okay, non-coding. So NC denoted as non-coding RNA. There is no any coding, codon. It do not indicate any enzymes. Amino acids. Okay. The messenger RNA is also further divided. Housekeeping ncRNA, non-coding RNA. Again, the non-coding RNA, housekeeping non-coding RNA also divided into 2 parts. That is called as tRNA, rRNA. So here the 3 RNAs comes. What are the 3 RNAs? mRNA, tRNA, rRNA. On the other hand, the non-coding RNA is also divided. Regulatory ncRNA, non-coding RNA, it may be divided into 2 types. INC RNA, so long non-coding. LNC, long non-coding RNAs. This is what is that? Equal to 200 or less than 200 nucleotide per NEET. More than 200 means what is that? Small ncRNA. So the microRNA, snRNA, siRNA, snRNA, piRNA. So these are the examples. These are the diagram. Okay. And also piRNA. So how many RNA is there? Different types of RNA have been reported by the scientists or discovered. But we need only the 3 RNA molecules only. What are the RNAs now? rRNA, tRNA, and mRNA. These are the 3 components only essential for the protein biosynthesis. See the next one? You can understand. See, these are the 3 RNAs there. RNAs, this is called as mRNA. mRNA is nothing but messenger RNA. tRNA, so otherwise called as transfer RNA, and also called as adapter RNA, otherwise called as a clever leaf model RNA, like that it has. And also it is a 2D structure, 2-dimensional structure. tRNA also exists in 2 forms, 2D structure and 3D structure also. 3-dimensional, 2-dimensional structure only present in the tRNA molecules only. And also adapter molecules, tRNA is also called as adapter molecules. Why it is adapter molecules, we will see. Okay. And also the rRNA is called as ribosomal RNA because it's made up of ribosome. Already you know about ribosome is essential for the protein biosynthesis. Otherwise, it is called as a protein biosynthesis factory. The RNA molecule it has two subunits. One is called as large subunit, and also another one is called as small subunit. Okay, see that one. The RNA, ribosomal RNA types. See that how many types is there? 28S, 18S, 5.8S, and 5S. Abundance, how much amount of 80% of the total. This is rRNA. Don't forget, my dear student, this is very, very important. 80% present in rRNA only. Very stable conditions, very, very stable nature. The messenger RNA, see that one? So 10.5 different species. Okay, but here 2 to 5% only. Don't forget, my dear, 2 to 5%. Total 80 plus 5, 85. Okay, unstable to very stable. Unstable to very stable. The last one is called as transfer RNA. It is a 60 different species. This is present in 15% of the total. So total 100% it comes out. 15, 5, 20, 20 plus 80, 100%. Here so many times they were asked the question in this part, my dear students. This is also very stable in nature. Don't forget, my dear, this is very, very important. Ribosomal RNA, messenger RNA, transfer RNA is there. Which one of the following RNA is most abundant in nature? That means 80% of the total is rRNA, otherwise called as ribosomal RNA. Which one is the least molecules, least RNA molecule present in it? Like that, they may ask the questions. See that mRNA, see that mRNA is a single polypeptide. So this is 5% only. tRNA, how much is that? 15% only. 2 subunit is that. This is 80%. So 5, 15, 80. So total 100% present in it. Very stable. This one is also very stable. mRNA, it is unstable to very stable. Okay, and these are all are small RNAs. Some of the small RNA have been reported. Smaller nucleus, that is denoted as SN RNAs. This is also present 30 different species, but here less, more than 1% or equal to 1% of the total. It is also very stable. miRNA, microRNA, 100S to 1000 species. That is also 1%, more than 1%. Total, okay, stable. So here in this slide, you must note these three is very, very essential. rRNA, mRNA, tRNA. Percentage, rRNA 80%, mRNA 5%, transfer RNA is 15%, total 100% is very, very important. Now we are going to see one by one. Ribosomal RNA is the first one. Ribosomal RNA is made up of ribosome. The ribosome is it has 2 subunits. It was first discovered by the name of the scientist called as Kunz, Kuntz, ribosomal rRNA, discovered by Kunz. Okay, don't forget, take notes, my dear. So see that structures. This is the ribosomal RNA. So this linear structure is called as what is that? mRNA. The ribosome RNA is also called as a protein factory, protein synthesizing factory, or factory of protein biosynthesis. The ribosome RNA consists of 2 units is called as 2 unit is there. One is a large subunit, another one is called as small subunit. The large subunit as well as the small subunit united together. Okay, in prokaryote as well as the eukaryote, the ribosome will be vary according to this structure. Do you know prokaryote which type of ribosome present in it? In eukaryote, which type of ribosome present in it, my dear? Ajay?
So,
Yeah.
not sure, sir.
Okay, okay, anyway, doesn't matter. We will understand. See that? See the structure. So you can easily identify. See that one? The eukaryote ribosome, they told 80S ribosome. What is that? 80S.
So 80S, sir.
Yeah. Suppose if it is a prokaryote, see that? 70S.
70S, sir.
Yeah, don't forget, my dear. Okay. But
Yes,
division will be differ. Division will be differ. Just imagine this is the prokaryote nature. Here they have given us a 70S ribosome present in it, but the large subunit nature is 50S. Here the 30S. Totally you calculate is 8S it comes, isn't it? See here, I will show you.
Yes, sir.
Yeah, I will show you how it will be, because I have to move on to the whiteboard. So then only it will be better. See that one? Take an examples. A ribosome. Okay. The ribosome already in our board. So this is called as mRNA. This is a mRNA. Okay. Another one is called as, what is that? Ribosome RNA. Ribosome RNA. It has a 2 subunit. It has a 2 subunit. See, sorry, this one I have to put large one instead. Okay, see here. Okay, see here. So this is the, okay, this is the this is called as RNA. This is a ribosomal RNA. See that one? This is a ribosomal RNA. See here, the 2 subunit is that. This one is called as large. You know very about this one is called as large subunit. This one, large subunit. This one is short. This is a small subunit. This one is called as small. See that one? Small subunit. This one is called as, what is that? Large. Okay. But in prokaryote and eukaryote, the ribosome RNA, it will be differ. Already I told you. The prokaryote, what is that? And eukaryote, how it will be. And take an examples of prokaryote. Okay. Prokaryote, how much it contains? 70S there. 70S. 70. Yes. Okay. The 70S. And eukaryote, just imagine. So how much is there? No? 80. Yes. Don't forget, my dear. So this is very, very important. But the division, how it will be, the 70S, it will be divided into, see here. Here, how it will becomes, 50 and 30 in prokaryote. Total you will get 80, isn't it? Total will get 80. This is for prokaryote. But why they have given us a 70S? See here in this part, note it down in this part, in this part is completely reduced. From here, 5 minus. It is overlap with each other. Here also minus 5. When two round is overlap with each other, what happened? Just like this, isn't it? This portion is completely minus from the total one. So what happened? 70 yes only the final. Actually, this is 50 plus 30. 80 is there. In this portion, minus 5 minus 5. From the small subunit, minus 5. And large subunit is also minus 5. You will get, how much is that? 70S. Okay. Here 25. Here also 45. So total 70. Likewise, that eukaryotic also, how it will be divided? 80 is there, but they divide into 60 into 40. In this portion is there, 60 into 40 like that. The upper part is 60. The lower part is, what is that? 40 is nature. But in this category, they minus, which one, large unit as well as a subunit. This one is large, this one is called short. So large units as well as small unit, they minus 10 in the large units as well as this one is also small units also minus. Okay, in this portion, completely removed. So as a result, 10 plus 10, 20, 20 minus from the 100, you will get 80s. Are you clear, Ajay, my dear?
Yes, sir.
Will you please tell me, so prokaryote, which type of RNA present in it?
Sir, prokaryote 70s, sir. Eukaryote 80s, sir. The
Okay.
division in prokaryote will be
Hmm.
50 plus 30, sir.
Okay,
While
good.
in eukaryote, it will be 60 plus 40, sir.
Okay, very good, my dear. Exactly correct. So, okay, thank you. Good. See that one. So, we can move on to the slide. Okay. See that, my dear. You can see that this is a subunit, large subunit. This one is called a small subunit. Is that? See that it is the most stable form of RNA. The RNA constitute about 80% of the total cellular RNA. This is very, very important. More abundant. Which RNA is more abundant in the living cells or cellular or organism or any human body, whatever in your body, like that may they ask the question. 80% ribosome RNA, ribosome essential for protein biosynthesis. Okay. It is made up of 120 to 4,700 bases, nitrogen bases. Don't forget. The rRNA consists of single stranded twisted upon itself its some region. In the helical region, most of the base pair are complementary and are joined by hydrogen bond, because bonds are nitrogen bases always bind with the hydrogen bond only. The RNA is formed from only a small section of the nuclear DNA, and there is no definite base relations between the rRNA and DNA. Once again, 80% of the rRNA present in every organisms. How many nuclear base pair is there? 122, 4,700 base pair present in it. But the nitrogen base pair or complementary genes are united together. Which bond? Hydrogen bond. How many molecules, how many nuclear DNA molecules require means a small section is enough for the productions. See that one? The large subunit, already we discussed, 60S of 80S ribosome contain 3 molecules of the RNA. Don't forget, my dear. The large subunit also, it contains 3 molecules of RNA having sedimentation coefficients of 28S, 5.8S, and 5S. You have to calculate in this manner. 5S, 5.8S, and 28S. So these are the 3 subunits present in where, the 60S RNA. Of the 80S. 80S means eukaryote. Okay. And the small subunit, it contains 40S. Because 60 plus 40, 100. 20 minus already in our board. It has only 1 rRNA molecules of 18S. You can see the structure. You can clearly understand. See the structure? So this is the structure of eukaryotic ribosome. It has 80 S, otherwise called a Swedberg unit. This is S is called a Swedberg unit. Swedberg, SWEDBERG, Swedberg unit, otherwise called as sedimentation unit. Okay, 80S. Where? In eukaryote. The 80S divided into 60S and 40S. The 60S subunit, it consists of 3 RNA molecules. See that 3 RNA molecule is there inside. 5, 5.8 and 28S RNA. At the same time, remaining 40S subunit, it contains only the 18S RNA only. Are you clear, my dear Ajay?
Yes, sir.
Yeah. So don't forget, my dear, here so many times they were asked the question. So many times. Okay. The eukaryotic ribosome, the 60S subunit contain how many molecules? Which one is the add one? Like that they may ask the question. Suppose the 18S also given in this part. 5 and 5.8 and 28. What 48 subunit contain which one of the following? 28, 5.8, 18 is like that they may give. So you have to clear 40s only present, 18s only. 60s it contain 3 types of molecule. That is named as a large subunit contain 3 RNA molecule. Small subunit contain only 1 sRNA only. Okay, don't forget, my dears. And then the large subunit 50s of 70s ribosome. This is called as prokaryote. The ribosome, it is mostly found in the prokaryote. Bacteria has 2 RNA molecules having sedimentation coefficients of the 23s and 5s. The 5, 5.8, 28. But here 23s and 5s only. The smaller subunit 30s has just only RNA molecules of 16s. There 18s. Here 16s. See that structure you can easily understand. This is called a 17s ribosome. That is 18s. Here 17s. The 17s subunit, I mean ribosome, the large subunit contain only 2 RNA molecules only. What are they? 5s and 23s. So before that one, the prokaryotic ribosome is 70s, right? The 70s is divided into 2 subunit. One is called as large subunit and small subunit, right? The large subunit size is 50s subunit and the small one is 30s subunit. Okay. The 50s subunit contain only 2 RNA molecules only. Okay. The 5s and 23s. But 30s subunit, it contain only 1, similar to that of the 80s ribosome. Okay. You can see the next diagram. You can easily compare with to each other. So this is prokaryote. This is eukaryote. Prokaryote ribosome 70s. Eukaryotic ribosome 80s. Okay. It has two subunit. It is also has the 2 subunit. Okay. How it will be divided? 70 divided into 50s large subunit, 30s small subunit. This 8 is divided into 60s and 40s. Okay. Here, how many RNA molecules present in prokaryote? 2 RNA molecules. 5s RNA, 23s RNA. Okay. Here, the large subunit divided into 3 RNA molecules. 5s RNA, 5.8 s RNA, and 28 s RNA. So here the 3, here the 2. But the small subunit, only one variation is here. What is that? Here 16s is there. Here 18s is there. Are you clear, my dear Ajay, in this part?
Yes, sir.
Yeah. So in this part, don't forget here the question, each and every question, every NEET question, they are asking. Don't forget, my dear, practice it once again. 70s, 80s, 50, 30, 60 and 40. 2 subunit, here 3 subunit. 5, 23. 5, 5.8, 28. Here 16s, here 18s. Okay. So this is a very, very important, my dear. See that? The entire process. This is the ribosome RNA. This is the messenger RNA. This one is called as messenger RNA. See that diagram. This is a tRNA. tRNA, it has a different sites. It can bind. We will see. Okay. So in the protein synthesis. Next. First we studied rRNA. How many percentage present in it? 80%. Don't forget. Next, we will pass on to the next RNA is called as transfer RNA or tRNA, otherwise called as adapter molecules, adapter RNA. Okay. See here, this is first postulated by the name of the scientist called as Watson Crick only. Crick postulated the presence of an adapter molecule that would read the code on one hand and would bind to the specific amino acid to the other hand. That's why it is called as adapter molecule. Why it is called as adapter molecules means the tRNA, it has a 5 arms. But here, one hand, what is that? It would read the code on one hand, and would bind to the specific amino acids the other hand. That's why it is called as adapter molecule. Okay. It is the second most common RNA in the cell, and it is called as soluble RNA. Don't forget, supernatant RNA, otherwise called as sRNA, otherwise called as soluble RNAs, sRNA, and also called as adapter RNA. Don't forget, my dears, the tRNA is also transfer RNA, soluble RNA, supernatant RNA, and also adapter RNA. It has a different names, tRNA. Okay. So don't forget. It was discovered by the Hagland. Okay. So tRNA was discovered by Hagland. It is the smallest in size and made up of 73 to 93 nucleotides. Don't forget, they ask the question tRNA, other name. tRNA or mRNA or rRNA, it consists of how many nucleotides? What about the molecular weight? And then third one is how many percentage of the total cellular content? Like that, they may ask the question, my dear. Don't forget, in this part, it is the smallest size, so the number of molecules also very less, 73 to 93 nucleotide. Okay. And then it is molecular weight range between 25,000 and 30,000. And the sedimentation coefficient is 3.8s or 4s. This collectively 3.8, they may be calculated as a 4s. It constitutes about 10 to 15%. Already you know about that is 80 tRNA, 15%, and rRNA 80%, and mRNA only 5%. Already you know about of the total cellular RNA. And one more is that there are more than 60 types of tRNAs. 60 types of tRNA also identified in different organisms. This is also another one. So in this category, tRNA and rRNA is very, very important. Don't forget, my dear students, in this part. Okay. There are more than 60 types of tRNAs, different of length of the D and V arms. Okay. So what is meant by arms? Okay. There are 5 arms is there. You will see the structure of the tRNA, then only you come to the conclusion. See that this is the structure. Okay. And also called the clover leaf model. Clover leaf model. Already you know about that rummy circles. You know that. Okay, the clovers. So having the structure look like a clover leaf. Clover. See the 1, 2, 3. So in this part, the 5 arms is the first in this area is called as acceptor arms. This one is called as acceptor arms. The another one is called as T loop, otherwise called as T arms in this part. So in between T loops and also anticodon loops, there is a small protrude-like structure. This is called as variable loop. This is anticodon loops. This one is called as D loops, otherwise called as dihydroxyuridine loop. So total how many arms is there? In D, tRNA, acceptor arms, T loops, variable loops, anticodon loops, and D loops. Okay, my dears, don't forget. So these things we will go discuss one by one. See that one? It contains many unusual bases, 15 to 20% like inosine, pseudouridine, and dihydrouridine. How many percentage of the unusual bases present in it? Like that, they may ask the question 15 to 20%. How many percentage of the tRNA present in the cellular activities? 15%. Okay. Likewise, unusual bases also 15 to 20%. Okay. See that. Which component like as a inosine pseudouridine and also dihydrouridine. Okay. These are the unusual bases. Identical structure in prokaryote and eukaryote. Thymine is present unusually. See that the nitrogen bases of the some nucleotides 15 to 20% get modified so that single standard tRNA becomes coiled into clover leaf like a form, that is 2D structure. The clover leaf model is also called as 2D structure, 3D structure, 4D structure. Like that, they may ask the question. Clover leaf model only 2D structure. They may exist in another form. Which one? An L-shaped form is also there. Which one? tRNA also it exists in L form, that is 3D form. Sometimes they may ask the question. The 3D form of tRNA is also called as L form, M form, cloverleaf form. Like that they may ask the question. This is very, very important, my dear students. 3D structures L-shaped. 2D structure cloverleaf model. Otherwise called as normal DNA molecule. See the structure, you can understand. This is the cloverleaf model. This is the 2D structure. Okay. Already I told you how many arms is there. Acceptor arm, T loop, variable loop, anticodon loop, D loop is also. Maximum, this question is, what is that, Diagrammatical question is. Okay. They may ask the question diagrammatically. So many times they were asked the question. Which part, this part, variable region. This is also very, very anticodon regions. This is very, very important. Dihydroxy and also the acceptor arm. Acceptor arm, anticodon arm, D loops. So in some cases, they may ask the question, D loop, this part. The anticodon loop is very, very important. Without anticodon loop, there is no any protein biosynthesis. It will not take place in the ribosomes. Okay. See that one. The nucleotide sequences, primary structures of the tRNA was first worked out by Hawley et al. 1965 for yeast alanine tRNA. Primary structure. Okay. This was first discovered Hawley et al. Et al is nothing but the co-worker. For his extraordinary work, Hawley was awarded Nobel Prize in 1968, which he shared with the Nirenberg and also Corona. So these are the co-workers. These are the co-workers. Et al denoted as Nirenberg and also Corona. Several models of the secondary structures of the tRNA have been proposed, and of this cloverleaf model is the most widely accepted. So many times scientists, they have given its own specific model. Okay. Particularly, which one is the accepted worldwide means the cloverleaf model is worldwide accepted one. Okay. See that one? This is very, very important, my dear students. See the structure, you can easily identify. This is the cloverleaf model. How many arms is there? 5 arms is there. Acceptor arm and T psi C arms. This is called as T arms, T loops, they told, isn't it? But exactly what is that, T psi C arms. Okay. This is also a extraordinary anticodon loop. DHU arms. This is called as trihydroxyuridine. You can see the next diagram. Can understand, see that structure. 2D structure, 3D structure. See that. So this is the structure of 2D. This is the 3D structure. Okay. So these are all are important. We will see in the deeply in the next classes, but we must know what is by tRNA, how many types. See. Okay, my dear. So we will see the remaining things on the next classes. I will summarize all those things. Okay. So in this part, what already we discussed, types of RNA molecules. How many types of RNA molecules present in it? There are several types have been identified in the chart. Particularly, there are 3 molecules is very, very essential. One is called as mRNA. It mostly found in 5% only. tRNA, it is a 15% only. rRNA is the 8% most abundant or more abundant. It's very, very important things. Remaining things also divided into micro, SNO, SI, and SN and PI. Okay, so these are the things. Particularly these 3 is very, very essential. mRNA, messenger RNA, tRNA, transfer RNA. So many things is soluble and insoluble like that. Okay, and rRNA is also there. Okay, so different types. Okay, see that one. Ribosomal rRNA. How many molecules present in it? How many molecules? mRNA, how many things is there? tRNA, how many things is there? It is also how many percentage? 80% abundant, 2.5%, and 15%. Stable or unstable? Sometimes they may ask the question. This is also just to give a glance. Okay. The ribosomal RNA, Kunz already know about. It consists of 2 subunits, large subunit and small subunit. It may be vary in the eukaryote and prokaryote. So that 80% of the total cellular RNA is the rRNA. Already know how many molecules nucleotide present in it. 122 is 4,700 base pair is there. The bond is also hydrogen bond. Okay, small portion is enough for the synthesis of the DNA molecule. And then how many molecules? 68 ATS. How many subunits is there? There are 2 subunits already know about. ATS. Okay, it has a 2 division is there. Excuse me. 60S and 40S. It consists of 3 subunits, 5S, 5.S, 8S, and 28S subunit is all present in the large subunit. Here, the only 8S is the same structure is also present in the prokaryote structure. It is also 70S ribosome present in it, 50S and 30S. But here, the 2 ribosome only present in 5.S and 23S RNA. Here, 18S RNA molecule is present in it. So the comparison already know about how it will be. Okay. And then, so this is the entire structure of the protein biosynthesis, otherwise called as protein factory. And transfer RNA, already we saw it. It is here, you must note, who stated the statement, it's Crick. And it is also called, what is it, soluble RNA, supernatant RNA, or also soluble RNA, and also adapter RNA molecule. This is very, very important, my dear. So why it is called as adapter RNA? Already we discussed. One hand is, what is a reading or the codon, and another hand is the what is the specific binding of the amino acids. Okay, which is the how many nucleotide is there? 73 to 93 nucleotide. Who discovered? Hagland. How many molecular weight is there? 25,000 to 30,000. The sedimentation coefficient, it's exactly near to the 4S or 3.8S exactly. How many percentage? How many types of RNA molecules discovered by the scientists? What are the types? D and V arms. Okay. And also how many percentage of the unusual bases present in it? Already we discussed. The nitrogen bases of nucleotides, so 20% is there. 2 types of molecules, 2 types of structure have been reported. One is called as cloverleaf model, otherwise called as 2D, and L-shaped forms, otherwise called a 3D form is there. So this is the structure of the tRNA. It consists of 5 loops, 5 arms is there, acceptor arm, T arms, variable arms, anticodon arms, and also D loops, otherwise called as D arms. Okay, so this was first discovered. Who is discovered? Already know about equally tied sequences. Coley et al. 1965. He got 1968 Nobel Prize. He chaired with the Nirenberg and Corona. What are the 2 types of model? Is that tRNA have been proposed, and the cloverleaf model is the most widely accepted model. Okay. So that's all about, my dear students, today this class. We will see the remaining things on the next classes, my dear. Thank you so much for the participant, Ajay. Thank you. Bye. Take care. Thank you, ma'am.
Thank you, sir.
Thank you, ma'am.